Okay, let's talk cellular respiration. Cellular respiration is something that happens in all cells to an extent, but the most efficient form is found in mitochondria and all like eukaryote cells. So the equation basically is 6O2 plus glucose is turned into 6CO2 plus 6H2O, and then a varying amount of ATP depending on efficiency of the whole thing. First, let's take a look at the first step, glycolysis. This is not found in the mitochondria itself, but in the membrane around it, right? So if you have a cell, this would happen in the cytoplasm. So this consists of glucose plus 2 ADPs plus 2 Ps, and then 2 NAD pluses. So this altogether basically breaks apart the glucose into two pyruvates, and the energy from this breakage creates two ATPs as well as a hydrogen and electron, which are stolen from that glucose and are brought over to the NADH. And this is like a taxi for a later step in the whole thing. And like I said, a pyruvate is basically one half of glucose minus a hydrogen proton, or and I'd like to say and an electron, right? Because for each NAD plus, um, a hydrogen and an electron are taken, right? Now, the NADH is carried to one of two places depending on if O2 is present in the cell. If it's not present, then this NADH is converted back into that pyruvate to create something like lactic acid or ethanol. And this is called anaerobic respiration, or depending on the type, like if it's with ethanol, specifically fermentation. This is pretty inefficient, but we'll get to that later. Now let's assume that you do have O2. Well, then the NADH gets to go to the ETC eventually, but not yet, and the link reaction takes place. So what is the link reaction? Basically, in the link reaction, the pyruvate that we had originally gets broken down even more into two more NADHs, acetyl-CoA, and some CO2 waste. And this NADH goes to the matrix along with the acetyl-CoA for later steps. And this whole thing is called aerobic respiration. Um, so eventually, these two separate reactions, aerobic respiration and anaerobic respiration, will lead to one of two possibilities. If it was anaerobic, it will only lead to a net gain of 2 ATP per glucose. But if it was aerobic respiration, it's 34 to 38 ATP per glucose. And that depends on the efficiency of the mitochondria and like the environments it's in. It could just get lost somewhere throughout the whole thing. But, keep that in mind. Whenever you have O2, it makes a cell wildly more efficient. Now, as mentioned, the two acetyl-CoAs get brought to the matrix of the mitochondria, which I'll get over the anatomy of a mitochondria later. But what we need to know is that it gets taken to the thing called a matrix, and it begins something called the citric acid cycle, or the Krebs cycle, and it's similar to the Calvin cycle, but basically in reverse. And you will be left with 2 ATP, 6 NADHs, and 2 FADH2s, right? You have 2 hydrogens and 2 electrons per FADH. So in total, you have 6 over here, 4 over here, so you have a total of 10 protons that get moved to the electron transport chain, but we'll get into that, as well as some more carbon dioxide waste. Now, this carbon dioxide waste, you might be wondering where it goes, and the simple answer is that you breathe it out, and most do, other mammals. That's why, if you pant, if you're, working out really hard, that kind of stuff. Um, and if you've been paying close attention, you might be wondering, well, like, where does all this glucose come from? It's not like these cells are finding it on their own. And the answer is GLUT4. GLUT4 is a protein that is found in your bloodstream, and it acts as a like a sugar Amazon. It brings these like sugar packages to all your cells so they can work, and it keeps your whole system running. And interestingly, GLUT4 is released in response to like insulin levels. So, if sugar is received by insulin, you know, that reaction will eventually send out GLUT4, which will then take that sugar that it just found, send it to those cells, and power them. And that's that. Okay, so here we have the inside of a mitochondria, right? Imagine I split it right in half. This is what it would look like. So first you have your outer membrane and you have your inner membrane. And in between these two right here, you have this thing called the intermembrane space. That's what this thing in white is, right? So you could imagine this as the mitochondria's cytoplasm almost. Now, inside of that, there's an even further thing called the matrix. And in this matrix, you'll find something called the electron transport chain. Now, just to recap, the citric acid cycle, link reaction, and glycolysis have been making a lot of NADH, right? And this NADH is floated into the matrix, right? It is brought over here and it is broken apart. It's simply broken apart into NAD plus and a single hydrogen as well as an electron. Now the electron actually powers the facilitated transport or the active transport even of this hydrogen proton through this. So it goes from low to high concentration into something called the intermembrane space, right? This thing right here. So imagine this thing right here is chock full of protons at all times if it's working efficiently. Now all those NADHs from all those separate reactions are moved over, and the same thing goes with FADH2, and that becomes FAD plus 2 hydrogens and 2 electrons. And once again, those electrons now push those 2 hydrogens into the intermembrane space. Now these hydrogens, they're bubbling around. They need to get out somehow. They need to get some out somehow. And the way they get out is through the ATP synthase, almost exactly like the same ATP synthase that you see in the photosynthesis electron transport chain. So now these hydrogens go down through this and they power or they give enough energy for ADP plus P to become ATP, right? And now these hydrogens that you find lying around are actually bonded with the oxygen. Remember the oxygen from the very beginning right here? This oxygen is really just used to carry away these hydrogens from the whole reaction, right? To maintain this concentration gradient. And similarly to the CO2, it is also breathed out as you're, exercising and stuff, but it's also used in other places. It might just be peed straight out, or for example, you could sweat it out. Water has a lot of uses in the body, and there's a whole reason why we're 70% water. So, it could be recycled later on, but that's where that comes from. And all this ATP that's made from all these separate hydrogens is enough to, in total, make aerobic respiration, cellular respiration that has oxygen present to create 34 to 38 ATP per glucose. Just a single glucose makes that many, which is quite a bit. Especially when you compare it to anaerobic respiration, which is only 2 ATP, right? There's a huge difference, in the amount of energy produced from this reaction, and it's safe to assume that anaerobic respiration is not very helpful, especially when you have a much bigger mammal. You know, that is why people die when they're underwater, because if they didn't die, then they would be really efficient with the 2 ATPs they get per glucose, and they have to spread it crazy thin. But unfortunately, that's not how the world works, and if you run out of air, you die, most often, usually. Now, there is two separate forms of phosphorylation that you should really know the difference of. There's something called substrate level versus oxidative phosphorylation. Now, oxidative phosphorylation is the electron transport chain, okay? That is the most simple way to memorize it. Even better, you think oxidative, oxygen, where do you find that? Electron transport chain. Now, the substrate level phosphorylation, that is actually, closer than you think. You can actually find that in glycolysis and the Krebs cycle, right? There is substrates and such, like everywhere throughout it, and just the breaking apart of these substrates is what powers the phosphorylation of ATP. Phosphorylation is basically adding a phosphate to something, right? That's where the phos part of phosphorylation comes from. So the idea is that substrate phosphorylation is that the energy comes from the substrate, while oxidative phosphorylation is from something else, like this pushing of hydrogens through the ATP synthase over here. There's also another term that you should probably know as you go forward with AP Bio or regular bio or whatever you're taking exactly, and that is the difference between oxidation and reduction. Now, reduction is reducing the positive charge of a molecule, which basically means that is gaining electrons. But of course, they didn't want to call it gainage or something like that because scientists are weird, but it's okay. The best way to think about it is that reduction is the reduction of a positive charge in a molecule. Okay, you're reducing that positive charge away from it by giving it all these electrons. Okay, now oxidation is the loss of those electrons, right? You're taking them back, you're taking them away, kind of how oxygen, how it's that, for lack of a better term, fat molecule, and it really hoards all those electrons from hydrogen, right? You could say that oxygen is oxidizing hydrogen because it's taken away those electrons, even if it's temporarily, right, through those covalent bonds. It's taking them away, and it is increasing the charge of the electron, making it more positive. So that's how you could think of it. Um, that's about everything you should know about cellular respiration. If you have any questions about photosynthesis per se, I would really watch my other video. Um, Hello, Felix. That's that's about it. Um, if you want to see these videos quicker, I'd recommend subscribing or something like that. So you don't have to wait for it to eventually show up in your recommended or something like that 5 days after you've taken the test. You know, I wish you the best of luck, and bye bye.